Thank you. So I'll say up front, we're currently not doing any eye therapy with RNA LNPs, but we're interested in doing this, and we're waiting for people to come to us and say, hey, let's try something. So my conflicts of interest, we've got a bunch of companies and a bunch of patents. So I want to start off, and I do this because I spend a lot of time talking to lay audiences. And one thing they're guaranteed to say to me is, I don't want to take the vaccine because it was invented in 10 months. And I try not to laugh because that's inappropriate. But RNA was first identified in 1961, and it was first injected into an animal in 1990, and it was in human clinical trials in mid-1990s. So it's not something new. When Katie and I started this research, I was still going to HIV meetings, and I would talk to friends like Tony Fauci and Gary Nabel and others, and would tell them what I was working on. And they would look at me and they would nod and they would smile. And then when I finished, they'd say, oh, that's interesting, Drew, but why don't you do something useful with your career? RNA is never going to go anywhere, and you shouldn't bother. And what I would argue is that protein therapeutics are the largest expanding element of pharmaceutical development. To make a monoclonal antibody, you start with a 50,000-liter drum of CHO cells or other cells. After that, you have to figure out how to purify that protein, and every protein requires a different purification. So for this reason, monoclonal antibody therapies are $50,000 to $100,000 a year. So that excludes most of the world, and it excludes a lot of the United States population. For RNA, RNA can be made in a small container. Moderna and Pfizer use 100-liter bioreactors that make more vaccine than a 50,000-liter drum of adenovirus-producing cells. There's no mammalian cells, there's no cells at all involved. It's an enzymatic reaction. It's identical for every RNA, so if you change the coding sequence, you don't have to change the production procedure. It's also safer. CHO cells and other cells sometimes misfold proteins. With RNA, our cells make the protein and our cells fold them correctly. So something that we've been working on for a few years is thinking about the future of RNA beyond vaccines. And what we've done is we identified, and many others have, the principal problem with nucleic acid and viral therapies are getting them to the right cells. So LNPs go to the liver and they go to dendritic cells. What if you wanted to target a different cell type? So we've been working on targeting a variety of cell types. These were the first three that we looked at. Our initial approach was to alter the lipid nanoparticle, to add a targeting ligand or targeting antibody to the surface of the particle. When we did that, the morphology of the LNPs didn't change. The EMs look identical. They're a little bit bigger. That's about the size of an antibody coating. And most importantly, the surface charge doesn't change. And why that's critical is because the surface charge determines toxicity. Lipid nanoparticles have a close to neutral surface charge. If it's positively or negatively charged, like other types of liposomes and delivery vehicles, that activates either clotting or coagulation. So neutral charge is maintained. Most importantly, they retain function. So in A, this is binding to a PCAM expressing cell line of a PCAM targeted LNP. And in B and C, we're showing after uptake that's mediated by targeting, we get translation of the RNA. So the lipid nanoparticles still work. When we inject them into an animal, we alter the distribution of the uptake. So this is a luciferase encoding mRNA. And if you give the animals luciferase substrate, they produce light. In the absence of any targeting or in a control IgG targeted, it goes to the liver. When you PCAM target, it goes to the lung. Lung has the highest distribution of endothelial cells expressing PCAM. And you lose about half of the liver targeting, which is likely because you're blocking ApoE binding. We've looked at CNS, which is as close to eyes as we've gotten so far. So all the residents in the room will know this story. And this was our fear of a Friday night when a new stroke patient would come in. And when they first came in, they would have a middle cerebral artery stroke. But that wasn't the fear. The fear was what happened over the next few days. And what typically happens is that the stroke expands. And here it's covering nearly half of the hemisphere. This expansion is because dead brain tissue leaks inflammatory mediators. That causes vascular leakage, it causes inflammation, and the stroke expands. So using work that our pharmaceutical collaborators developed, we put a thrombomodulin encoding mRNA. Thrombomodulin inhibits inflammation and clotting, and we targeted the LNPs with VCAM. VCAM targets the blood-brain barrier in settings of inflammation. And we used a model where we injected TNF-alpha into one hemisphere of the mouse's brain. And then we treated them or not with LNPs with thrombomodulin targeted to the blood-brain barrier. And when we treated them with this agent, we reduced vascular leakage back to control levels. So we got rid of all of the inflammation, all of the vascular leakage. When we looked at these animals, the size of the dead brain stayed the same. It didn't expand. We're looking at moving similar therapies into people. As an immunologist, my main interest is in lymphocytes. And T cells are a wonderful example, especially at Penn, where CAR T cells were put into the clinics in the first two FDA-approved drugs. There's a lot you can do in modifying T cell function. You can both make them better, you can make them better killers, or you can turn them off. You can make them regulatory T cells. You can put CAR molecules on them to allow them to kill. There's a variety of different modifications. The problem is that T cells don't have endocytic activity. If you give them free particles, they can't take them up. So our idea was that if we targeted T cells with an antibody that bound an antigen that endocytosed, that would take up the lipid nanoparticle. And that's exactly what we saw. In the absence of targeting, we had no activity in T cells. When we targeted CD4, we specifically had uptake into CD4 positive T cells. When we injected these into animals, the spleen lit up. The spleen has about 20% CD4 positive T cells. But what was more interesting is we re-imaged those mice after we removed all of those organs. And what showed up were lymph nodes. So these are para-aortic and inguinal lymph nodes that are lighting up after an IV injection of CD4 targeted LNPs. What that means is that the LNPs escaped from the circulation, went into the tissues, into the draining lymphatics, back to the draining lymph nodes, where they were able to target CD4 positive T cells and express their protein. Here we just purified the T cells to show that the luciferase was in the T cells. We put these into a LOXP mouse. And what we saw were very high levels. So the FITC is the green color driven by the LOXP, had high levels in both spleens and lymph nodes. But what's more interesting from an HIV point of view, the y-axis is an activation marker. And we see that we had gene editing at similar levels in resting CD4 T cells. That's important because the HIV reservoir is in the resting T cells. This tells us that we can now target those cells to attack proviral HIV DNA. And those studies are in macaques right now. We had very high levels with optimization, around 50% of the CD4s in lymph nodes and spleen could be targeted. So these were our first studies of lymphocytes that told us that we could effectively target T cells in vivo. The range of things that you can give are listed here, but any kind of gene editor, any kind of protein, it's really, the potential is enormous. So everybody here knows about CAR Ts, so I don't need to go through this slide. But we wanted to know, could we make CAR T cells in vivo? Currently, for Chimera and the other CAR T drugs, you take cells from a patient that you leukapheresis, you infect them with a lentivirus, you expand them for about 10 days, you give the patient chemotherapy, and then you give the T cells back. That costs almost $500,000 a dose. It works wonderfully. The success rates are in the 60 to 70% range, but it has limited distribution. You don't find CAR T sites in sub-Saharan Africa, in much of Southeast Asia. They're just too expensive and too difficult to set up and run. So our idea was, could we make CAR T cells in vivo? The first thing we had to do was target all T cells, not just CD4s. So we used a variety of markers. This is the data for CD5. CD5 is a scavenger receptor on T cells, which means that it's endocytosed after it's bound. And what we found was that a single treatment had 80+% of the T cells targeted. So very efficient delivery. The idea for in vivo CAR T cell generation is that you target T cells, you encode the CAR molecule in the mRNA. The LNPs are taken up, the RNA is translated, and the T cells become CAR Ts. We use the CAR T directed against FAP that recognizes activated fibroblasts in a model that John Epstein set up of cardiac fibrosis. So this is an echocardiogram from these animals. And you can see in gray, these are normal animals. In beige, these are hypertensive animals that have fibrosis. All of the echo measurements show dilated hearts and poor ejection fractions. In red, we treated them once with CD5-targeted FAP-CAR LNPs. We returned cardiac function back to normal. So a single treatment returned the heart function back to normal. When we stained the heart for fibrosis, we could see high levels of fibrosis in the hypertensive mice that have returned mostly to normal with a single treatment. The other interest we've got are bone marrow stem cells. This is just a partial listing of all of the diseases that currently have treatments in generation. Most treatments for bone marrow diseases are bone marrow transplants, but we know they have a decent mortality rate, require lifelong immunosuppression, not ideal. A number of lentiviral gene therapies, ones were just approved for sickle cell anemia. But what you have to realize is that That requires you to take out a large amount of bone marrow from a patient, culture it with lentiviruses that edit the DNA, give chemotherapy to make space, and then put the edited bone marrow stem cells back into the patient. We don't have a price for that yet, but it's likely to be $2 to $4 million per treatment. The problem is 300,000 people a year are born with sickle cell anemia. They're mainly in sub-Saharan Africa, but they're present throughout the world. You can't afford 300,000 times $2 million, especially in sub-Saharan Africa. So our idea is that if we can target bone marrow stem cells and edit them in vivo, that will give us an inexpensive and easily administered therapy to treat sickle cell and all the other bone marrow stem cell diseases. This just highlights how you identify stem cells. In mice, it's a series of analyses. What's important is you start with 700,000 cells, and you end up with less than 1,000 repopulating bone marrow stem cells. They're incredibly rare. So that makes targeting difficult. We initially chose to use CD117, which is the c-KIT receptor, and we were able to link anti-CD117 to the LNPs, deliver these to mice. These mice their bone marrows have not been treated in any way. And we had very high levels, 80, 90, 100% delivery in vivo to bone marrow stem cells. We've treated Cre-Lox mice with Cre recombinase mRNA and followed them over time. And typically 70 to 100% of their blood cells became gene edited. Now, nobody believes this data unless you do a secondary transplant, where you take the bone marrow from a treated mouse, typically 6 months later, and you put them into a transplant model. And when we did that, 70 to 100% of the bone marrow stem cells were gene edited. So we had incredibly high levels of gene modification in the repopulating bone marrow stem cells. That's ideal for a disease like sickle cell anemia. The problem with sickle cell is you have to correct about 25% of the stem cells. And that's why the gene therapy is difficult. You have to take out a lot of bone marrow. This way, with 70 to 100% efficiency, we should be able to correct 25% of the stem cells by either increasing fetal hemoglobin or turning the sickle defect into a Makassar defect. But there are many other things you can do to bone marrow stem cells. You can deliver toxic proteins and potentially use this as an alternative way of clearing out the bone marrow when you're doing a bone marrow transplant. Mortalities from current bone marrow transplants range up to 5%. That's due to the high doses of chemotherapy that are required. Delivering targeted LNPs that can selectively kill the repopulating stem cells could be an alternative. You can deliver therapeutic proteins for a variety of diseases. You can correct gene defects. Once we get past sickle cell, there's thousands of other genetic defects in stem cells that we're looking at fixing. So we now believe that in vivo gene therapy can easily be done. The advantages beyond not having to have fancy, expensive hospitals to do this, the price is likely to be logs lower. We don't know for sure, but my dream is someday to be in sub-Saharan Africa with an igloo cooler full of RNA LNPs, giving people injections and curing their sickle cell anemia. And I think that's a very possible thing that will happen in the near future. So I need to thank all of the people who did the actual work, and I'll take questions.
Fascinating. Open for questions. Yes, Josh.
Drew, thank you so much for joining us and presenting this incredibly groundbreaking work. So for therapeutics for eye disease, some of the things we would wonder are, first of all, how long can mRNA expression last and how tunable is that length of expression?
Yes, so the difficulty is length of expression because we haven't looked at retinal cells, we haven't looked at other cells in the eye. Typically, they're translated for about 36 hours. So then it becomes a function of the half-life of the protein. But for gene editing technology, you want a short duration. You don't want Cas9 or base editors hanging around for a year.
And how efficient is the base editing that you've been working with so far?
So base editing is very efficient. RNA is a great way to deliver. Prime editing, I think everybody will say, is much less efficient and is not ready for therapeutic prime time yet.
So you can deliver the Cas9 plus the guide RNAs and get very very efficient correction,
Yep.
mutation correction.
Well, we've had CCR5 knockout in the 85 to 90% range.
Fantastic. And how about inflammation? So injecting LNPs into the eye is probably going to provoke some inflammation. I think you mentioned that charge is important. What else can you tell us about that?
So we and others are working on variants of LNPs that don't have adjuvant activity. We've identified a few that we're now using for autoimmune vaccines. It's technology still being developed, but ultimately I think that's what you want to use.
Wonderful. I don't want to hog the mic. Please ask more questions.
Cesar.
Really inspiring. And I'm curious, since you got your start in some ways in the world of HIV and going to meetings, what are the implications of these technologies in that realm?
So we've got a bunch. We've got macaque trials that are ongoing. We have a human clinical trial. We can deliver broadly neutralizing antibodies with mRNAs. Ted Kreider in my group has done 3 at the same time. We can make HIV-specific CAR Ts in vivo. We can deliver proviral editing enzymes that either cut out or inactivate the provirus. And Ted has made a new LRA, an activating agent, using CD4-delivered TAT that can turn on HIV latently infected cells and allow them to be killed. So the best part is all of that can be given as just an IV injection of LNPs, so it can be done anywhere in the world.
Over here.
So your cardiac research, where you're talking about cardiac fibrosis, I can look at sort of orbital diseases, thyroid eye disease, where you have fibrosis. When you got the reversal of the fibrosis, was it from just inhibiting inflammation, or what caused remodeling of the heart wall that actually restored normal function?
Yeah, so I think a lot of this is we didn't really understand the physiology of fibrosis. Originally, we thought you lay down collagen, it's there forever. That's not true. It's more like the bone, where it's constantly being remodeled, taken up, and laid down again. So we killed the activated fibroblasts that were responsible for laying down the collagen. And when we did that, it disappeared. Depending on the clinical situation, it will come back. Maybe you have to treat every 6 months or a year, or at some point, if you can't fix the clinical etiology that's causing it.
Yes, here in the back.
To know your thoughts and experience on growing multiple mRNAs. This is connected to multifactorial diseases, where you can replace with multiple mRNAs. So this thing is possible to cover that in any disease.
So we've made vaccines that have 20 RNAs in them, and we make potent responses to all 20 proteins. We routinely deliver monoclonal antibodies as 2 RNAs. We've delivered other therapeutics with, I think we're up to about 6 RNAs for therapeutic proteins. You get a reduction in the amount of protein made the more RNAs you deliver, so it becomes a balance. But you can definitely deliver multiple different proteins at the same time.
Well, I'm really glad you finally found something useful to do. What I'm wondering about, when we think about targeting aging-related neurodegenerations, including age-related macular degeneration and things in the eye, but also in the brain as a whole, in many of these diseases, we're targeting post-mitotic cells in which the endolysosomal system has been compromised. And I'm wondering whether we need to design the LMPs in a different configuration to help with that endosomal escape that is so necessary for delivery.
Yeah, I think that's going to be more of an efficiency. I mean, T cells have very poor endosomal activity, but when we were able to target them specifically, we got decent levels of translation. It was still a log lower than what we see with dendritic cells. So I think it's a matter of trying. The endosomal release is a function of the pKa of the ionizable lipid. So there's lots of things that could be played with, but I think it's just a matter of trying and optimizing.
That's encouraging. Thank you.
Yeah, thank you. Fascinating. I had the same question about the multiplicity of the LNP particles. So in addition to multiple molecules, is it possible to target like multiple cell types? Because apparently, complex neurological disorders in the brain or in the eye involve multiple molecules and multiple cell types. So can we conjugate like different antibodies like to the LNP particles to target like multiple cell types?
Well, we can and we have. My guess is we're not going to want to do that for multiple cell types because one antibody might bind at a higher efficiency, and we may only target one. But we can deliver two sets of LNPs, each with a different targeting marker to target the different subsets.
Thank you. Another question is about the toxicity. So if we apply or deliver the LNPs to the brain or to the eye, do you think like they will eventually like end up in the like leak out of the brain or the eye and then end up in the liver, for example, to cause toxicity?
Yeah, so the blood-brain barrier is pretty good at keeping particles out. So unless there's damage to the blood-brain barrier, I don't think anything is going to get out. Nothing gets in without specific targeting. For the eye, there I'm less sure. I mean, if they can escape the orbit and make it into the draining lymphatics, certainly. How much leaks out will be the question. If it's 1%, you'll likely never see that in the liver and you won't care. We also, so when we deliver CAR Ts, we don't care if the liver expresses a CAR molecule. And when we do gene editing, we don't care if we fix SCID or other genes or beta globin genes in the liver. It doesn't matter. We care when we deliver toxic proteins that we don't get expression in off-target organs.
Great. If we could thank Drew one more time. I'm happy to say that as of right now, my lifelong dream of both speaking in and moderating a session with a Nobel laureate has been fulfilled. Thank you, Drew, very much. Thank you to all of our speakers in this session. It was really fascinating. We're going to have a short break now.